expanded my coffee Spanish vocabulary. Thanks. Um, and so if, again, we already mentioned about sanitizing, but the lowering your microbial load, decreasing your temperature, and it has a note here about increasing your lag phase. And if you remember, that lag phase is kind of getting your microorganisms going in terms of their growth and development. It might increase the lag phase, but it also might keep your fermentation from kind of getting out of control. And that's where what I've found is, when we don't use water to kind of wash the coffee or to float the coffee or whatever when it comes in to the washing station, one of the biggest impacts I find is that it causes a really fast fermentation to get going. And if I'm wanting to use fermentation to develop flavors and develop characteristics, fast fermentations are not necessarily always your best friend. They're not necessarily always conducive to that. So by soaking or floating or using a hydraulic separation, we decrease that temperature and allow for a kind of a more controllable progression of microorganism development. The other control point is your water. And I think I mentioned this before as well, but I don't see a lot of research being done on water for coffee processing. It would be really interesting to do some research. However, the main point is because you most of the times people don't have any choice on what water they use. It's just whatever spring is nearby or river or whatnot. So, but your water is going to have its own microbial load. You're going to have, as we depending on the different mineral content and kind of chemical makeup of that water, you're going to have different types of dispersion of microbes and sugars, salinity of water. Yeah, absolutely. I mean, all of those different characteristics and aspects of water are going to be a contributing factor to quality. And like I said, I really do think this is an untapped area of research because of how significant it is in contributing to microbe development, accessibility of nutrients, accessibility of compounds that would then participate in those metabolic reaction or those metabolic pathways. Then one of the things we can control, so we're all still talking about our initial populations, those microorganisms, is inoculation. And in 2 lessons, we'll have a whole PowerPoint. I mean, Lalaman being one of the sponsors of this course, them being the yeast people, which I'm sure they probably would prefer a different nickname, maybe, I don't know. But there'll be a whole section. We'll talk a little bit about inoculation. But these can be commercial, or you're seeing a lot of people beginning to cultivate their own microorganisms based off their own environment and inoculating with those. It is important that when you are when you're not using a commercial product, it's a lot more difficult to maintain that the population thrives, that they're still alive, so to say. And so that's something that you just have to be you really have to figure out over time based off those specific, wild microorganisms that you're using. But it does, the idea with inoculation is that we're trying to outcompete any other or what other microorganisms are available. So where you can use inoculation with sterilization. So you've sterilized the environment, you've removed all microorganisms, and then you're introducing new ones. Definitely can be done, but more often than not, inoculation is being utilized where you're introducing a much higher microbial load, a much higher population into that environment in order to allow those microorganisms to outcompete any other bad or even maybe other positive microorganisms, but ones that you don't want to be involved in the actual process. And then backslopping, this is the aspect of where basically you take and producers have done this for a while. What they'll do is they'll typically take like a bucket or a cup or something of the fermenting liquid from one fermentation and dump it into the next fermentation. And it's kind of that same, it's a type of inoculation. The risk and it's something that can be done, and it's not, not necessarily bad, but the risk that it does carry is, let's say that fermentation batch, for some reason it encouraged or it saw the development of a negative characteristic, some microorganism that produced negative characteristics. If you then kind of introduce that microorganism into the next one, you've now contaminated that one as well. Yeah, like making sourdough. The trouble though is you don't know if the fermentation has gone wrong typically until the coffee is dried and you've tasted it, which could be 2 weeks later, or 3 weeks later, or 10 days later, depending on your own systems. So it is a little bit of a risk, but it is something that you could utilize to control the initial population. And then the final one is your tank material. And I mentioned about kind of stainless steel, but essentially, as you increase your porosity, which means the amount of holes, and even like microscopic holes, you increase your microbes. And so that's why things like, concrete that's not sealed, can be a place where microorganisms can live. And maybe that's a good thing, and maybe that's what is contributing to your really nice quality coffee, or maybe it's a negative thing because you have bad microbes living there. It's something to consider, and to be aware of. This one about being exposure to elements is always a big one for me. I always encourage that your fermentation tank should be out of the elements. Now, I realize it's not reasonable to build a warehouse for your fermentation tanks to go in, but at least you should have a roof to where you can keep the sun exposure directly off of, kind of keep the temperatures at least somehow regulated. But again, all of those are going to be contributing factors to your initial population. You're open in the environment, wind, rain, all of that kind of stuff is going to bring with it different types of microorganisms that are going to contribute and impact that fermenting environment. And this is why I was saying earlier, like, when you think about how coffee is processed and all these other factors that go into it, the fact that we're still able to produce really amazing coffees is quite incredible, and speaks to, I think, the hard work and ingenuity of producers across the world. Okay, so that's kind of when we think about how to control the fermentation, and we think about your microorganisms and that initial population. Those are different factors and different things that we can use to control and consider controlling those initial populations and what they are. Then we get to food. Okay, we used the term substrate, but food as being what microorganisms are consuming. And there's different controls that we can utilize or different areas that we can control to try to impact the different food. And then there's ways that we can monitor those. The first one we're going to talk about is your raw material maturation. Okay, so this is one of the first ways that we can control the types of food and the types of compounds that are available to microorganisms. Now, we're not going to talk extensively about how the coffee is grown, and I think another really interesting untapped area of research is soil management and fertilization, or whether that's organic fertilizer or whatnot, but basically soil health and soil management contributing to quality and flavor. But in general, we want ripe fruit to be used, and the consistent ripeness is critical when we look at the impact of our microorganisms developing flavor. And what we mean by that is the research that's been done and shown that the types of compounds that are available and accessible in unripe fruit versus that which is accessible in ripe fruit is very different, and therefore the food source for those microorganisms become different, and the byproducts that they could potentially produce. So having even maturation is not just so that you can take really cool Instagram photos with beautiful red, burgundy cherries and send it across Instagram. But it really is about having that consistent food source for your microorganisms as you begin your processing. There are a couple of different ways that we can kind of control that, or we can monitor that. Um, you can do kind of your, because how do you quantify? How do you measure maturation? There are ways that you can use, and I'll mention bricks a little bit later, and we'll talk about bricks briefly, but you can actually begin to measure the progression of compound development as the coffee matures. You see a picture here of a maturation range, and then you have kind of an evaluation, taking a sample and then counting how many fall inside or outside of that range. So there is a way to actually quantify your material. The tool, which tool, Hutan? Sorry, um, the board that's there with the. Sorry, I didn't follow which tool. If you want to explain, I'm happy to answer that. But what we can utilize different means, and it's always good if you're able to provide some quantification of that maturation level, so that you can essentially have a point to evaluate. If you're I'm always the kind of person that the more data, the more information you have, the better. If we can get more information and more clarity on what the starting place of that fermentation was, if something negative happens or even if something positive happens, we begin to better understand how to replicate that. The picture of the wood there, it's just like I've heard them called a maturation board, but essentially it's just a board that has 100 little kind of slots on it, and you take a sample of coffee fruit and you throw it on there, and then it gives you 100 coffee fruit that you can then count how many fall outside of or inside your target ripeness range. We call it a cherry board. Yeah, so I mean there's different terminologies for it. I've seen them made out of wood. I have a couple that are made out of plexiglass, just because it's lighter than wood when I travel. But yeah, no problem. We've mentioned this several times, but water becomes another huge contributor to your food source. So when we increase, we understand that sugar ripeness board. That's another good term. Another good term that we could use to describe those boards. Ibrahim, yeah, any of those would work great. Um, Anytime we understand, and we've talked a lot about fermentation, we know that glucose is a significant part of the food source in that process. When we use water, we're going to be diluting that. So fructose is soluble in water. So the more water that we use, the more, one of the impacts is the greater potential for diluting those sugars that then are no longer accessible to microorganisms in fermentation. So maybe the pulper that we would choose to use one that has less water. Or in transporting, pulped coffee from the pulper to the fermentation tank, instead of using water to do that, we might use a screw auger or gravity to do that, which would, utilize less water. There's different choices that we could make specifically on how much water we're actually using. Supplementation is this idea, and I started to see this in some of the coffee. So when we do the Q processing level 2 course, you do hands-on coffee processing, and the way that I've started to do it is basically teams get to kind of choose their own recipe, and I've seen people starting to use supplementation for giving additional substrate or giving additional food into the fermenting environment. So maybe it's coffee pulp. They're pulping a bunch of coffee and they're squeezing the juice out, and then they're introducing that. Or maybe they're putting fruits, or I have a good friend in Thailand. He just sent me some samples, and they put different types of fruits inside the fermenting environment to see the kind of potential impact that it could have. All of that is basically just giving and providing more compounds, more food for your microorganisms to consume to produce those metabolites. When you have maybe a stalled fermentation, introducing more of those could also be something that introducing additional food could be something you could look at. Is there any recipe of water quantity per kilogram of coffee for fermentation? No, not really. But the main thing that you would want to do is, if you're going to use water in your fermentation, you want to limit You probably just want to go right above the surface of the coffee mass. So you just barely kind of cover the coffee. That way you don't have extra water that could potentially dilute other types of compounds. And really it's just, it's wasteful. You don't need any more water than that. So um, typically that's kind of what the general recommendation is. Oops, there we go. All right. And then one of the other ways we could control this kind of food source is to do what we call in fruit fermentation. Okay. So in fruit fermentation, do they use third wave water? Like I said, coffee for water processing. It'd be like the new book. Anyways, in fruit fermentation, which we refer to as, what it very strictly says, fermentation when, before we pulp the coffee, so still in fruit. And you're finding more and more of this being done in coffee. And in fact, anecdotally, I can share with you that I have found, I have um, heard of, and I'm hearing more and more that people are finding more success in terms of um, inoculations when they're inoculating with a certain yeast compound or a certain microorganism. They're seeing greater impact in fruit fermentation inoculations than they are pulped coffee inoculations. Now, I have other friends, other colleagues who are utilizing pulped coffee inoculations pretty much exclusively. So, it's not necessarily one's better than the other. I'm just starting to hear more and more that there seems to be more positive influence or impact to in fruit fermentation inoculations than pulped coffee. But you do have in the skins of your coffee fruit, you do have polyphenols like anthocyanin, which is one of the compounds that contributes to the color change in coffee fruit. That can be a compound available to participate in these fermentation metabolic pathways. And so that could be a very different result and very different fermentation that could occur because of being in fruit, because the food source is different. And I said we would discuss Brix. The this being the way that we can measure or monitor this. Now, when we measure coffee, when we measure Brix, we are basically measuring soluble solids, and there's kind of this ratio of grams of sucrose per 100 grams of solution. Look, here's the thing about Brix. Is it solely measuring sugars? I'm convinced that there's probably some other compounds in it that it's measuring. It's not only just sugars, at least as we're talking about with coffee fruit. Now, Brix as a device and as a measurement is used in other fermentation industries or other industries that utilize fermentation to measure sucrose because they're specifically focusing on measuring that consumption of sugars during the fermentation process. However, I still think Brix personally is a valuable tool to help us understand and have a way to quantify what's happening. But I do not think Brix could be said, oh, well, my coffee was a Brix 28 and therefore it's better than yours, which is a 21. I don't see Brix as transferable in that sense. But to me, I use Brix a lot for establishing ripeness. So I'll take, green to black, and I'll take fruits from each of those kind of groupings and I'll measure the Brix and you can track when Brix begins to peak and help you identify and understand what your best ripeness range should be for your specific context. You can also take the liquid during a fermentation and you can monitor Brix as one data point, and you'll see the progression of that Brix number decline as those sugar compounds are being consumed. But that's one way that you can use Brix. This gives you a little bit of indication, like with red wine and American Pilsners and other things, like what different Brix levels correlate to within those, how they use those in those specific industries or those specific products. But really, there's not really any kind of recommendation for Brix levels with coffee because honestly, we're not necessarily, this is measuring essentially, these industries are focusing on it using sugar and specifically alcoholic fermentation that's converting that to ethanol, so that's producing alcohol, we're not necessarily concerned solely with that. One, because we're not consuming the liquid, but we're more concerned about other byproducts that are being produced. So I would say that yes, we can utilize Brix, and it's great to have that as a data point. But I would just be cautious putting too much stock into Brix as an indirect indicator to quality, because it's not as straightforward as what I think sometimes people try to communicate as they use that number. Okay, so we've talked about the initial population. So we're talking about how we control fermentations. We talked about initial population. We talked about the food source, and now we're going to talk about oxygen. Okay, now as we've discussed, last week with all the different types of microorganisms, the amount of those that thrive in oxygen, those that thrive in where there's no oxygen. Oxygen is a critical factor that we can control in order to monitor or control certain outcomes in fermentation. And each of these are different types of factors we can cover. We can use in our gases, and I'll discuss briefly about each of these here in a minute. You see our poor little oxygen molecule. In general, when we think about the impact to quality or impact to final characteristics, oxygen is going to be one of the major factors in selecting our types of microorganisms. Temperature is another one, but oxygen is going to be a significant contributor. Bacteria, if we have anaerobic conditions, meaning low oxygen or starved oxygen, it's going to favor your lactic acid bacteria over your acetic acid bacteria. For our yeast specifically, anaerobic conditions are going to favor alcoholic fermentation. Okay, so remember, your yeast are those facultative anaerobes that are able to function in both oxygen and no oxygen environments. Well, no oxygen environments are going to encourage more alcoholic fermentation, whereas oxygen rich environments are going to encourage the use of aerobic respiration, and so you're going to have different results from the different byproducts. For molds, your anaerobic conditions are going to restrict or inhibit mold formation. In anaerobic process, have we alcohol? Because I saw in Kenyan farmers drank that. Yeah, so actually, because of that being that low oxygen environment, you will have alcoholic fermentation, you will have yeast. And I saw it in Indonesia. We were doing a course, a Q processing, or maybe it was a Q Arabica course, I forget, but one of the participants brought a bottle of what was like a coffee alcohol, where they took that, they sterilized that, and they actually were looking at it as like another product. Personally, I didn't enjoy the taste of it, but it could be something that would could potentially be very interesting. So we one of the ways that we can monitor oxygen is we can actually ferment in a sealed container. Now, I don't know how well this video is going to play, but I'll play it right here. And you can see here in a second, you can see the top that exploded off the top. The percentage of alcohol, I'm not sure, Hutan. I don't know the numbers on that. Apologies, but I'm not sure exactly. I don't know if that video was clear or not, but basically what the producer has done is they've sealed that fermenting environment. There's been so much CO2 production that you saw the top like explode off whenever they were trying to release. Now, you will see most often people use these little valves, one-way valves. So I would encourage you that if you're going to kind of seal the environment, you're going to seal the tank, use a one-way valve. Yeah. Sorry, I don't know if that came through clear or not, Tristan, but so the amount of oxygen levels in that environment are going to depend on a few different things. One, the headspace. And when we say headspace, we're talking about, let's say you have a tank and you have coffee filled up to this level, and then you have the lid that goes here. That space between the lid and the top of the coffee is called the headspace. So if we want to reduce the amount of oxygen in that system, we can reduce the headspace where that oxygen can live, so to say. So what with these one-way valves, what essentially happens is as fermentation occurs, CO2 is produced and CO2 rises and pushes the oxygen out of the system. And you'll actually see where bubbles in these little one-way valves, you can see bubbles that begin to form and pass through that. The other way that you can remove oxygen is by pumping in an inert gas. Different types of gases can be used, but you can pump a certain gas in that then flushes the oxygen out. But during a normal or typical fermentation, you're going to have carbon dioxide production that's going to push the O2 out and create an anaerobic environment. So just by nature of sealing that environment and keeping away any additional oxygen, you're going to eventually create an anaerobic environment, an oxygen starved environment. Your tank shape is also going to impact your oxygen availability. You see the two shapes here, not necessarily that this is the only shapes that use, but something that is more narrow and tall is going to have very limited oxygen exposure, whereas a more kind of extended or very wide and narrow type of shape or tank geometry is going to encourage more oxygen interaction because you have more surface area that's going to be interacting with your ambient oxygen. How will headspace affect the fermentation? One, limited headspace is going to reduce the amount of oxygen that can survive in that system. So as the oxygen it kind of lives in that space, so you have CO2 that's being produced, but if you have a really large headspace, it's going to require a lot of CO2 to push the rest of that oxygen out of the environment. So it might take longer for that system to achieve kind of an anaerobic environment. Whereas if you have a very reduced headspace, it's going to be, it's going to take a lot less time for that CO2 to push it out, which then allows that fermentation to maintain that anaerobic condition for a longer period of time. So hopefully that helps. The other thing that we can control is submersion, so submerging or putting the coffee underwater. Now, there's a couple of things to note about submerging coffee underwater. And the first is that, yes, oxygen, so oxygen is available inside the water. We refer to it as dissolved oxygen. Now, it's not as high as the concentrations of oxygen in the air, but there is still some in the water. And what we can find is that during fermentation, that especially in a submerged fermentation, that oxygen is going to be used up. Okay. And you can actually buy a dissolved oxygen meter that will actually track when all of that oxygen is used up. It is understood that during a typical submerged fermentation, you are utilizing any type of dissolved oxygen, and so you are creating a low oxygen or oxygen starved anaerobic environment. So that's why there's sometimes consideration or sometimes talk the only way to do anaerobic is dry fermentation in a tank, seal the tank, and that's anaerobic. Or put coffee in a bag, suck all the oxygen out, and that's anaerobic. Well, when you have coffee that's fermenting underwater, you will also reach a point to where that system is anaerobic. Of course, it's going to be different, take different time, and you also have the water, right? So here we see some of the other impacts that water has is diluting sugar concentration or even decreasing the temperature. Water acts as a refrigerant, and so it's going to require a lot more energy to heat that to raise the temperature of that environment. So by using water, we can keep the temperature cooler for longer periods of time. Maybe that's positive, maybe that's negative. Depends on what you're trying to achieve. This note about homogenizing the fermentation conditions is the one that I typically hear of, is why water is utilized when people are inoculating, because it's easier to homogenize the entire system when you have water that's present. And so people that I know that are big proponents of inoculation, especially pulped coffee inoculations, they typically speak of the reason why they like to use water is because it homogenizes more of the environment or the system during that process. And as I shared, one of the ways to monitor, specifically your oxygen is you can use a dissolved oxygen meter. You can also look at the bubbling of that airlock as an indicator of CO2 production and pushing the oxygen out of that environment. But then we can also monitor some of the byproducts. If we're getting lactic acid or even if we're getting ethanol or alcohol, either aromatic or even in the taste, we understand that those are some of the byproducts of a reduced oxygen environment. And conversely, acetic acid, vinegar, being kind of those indicators of oxygen rich environment or oxygen rich fermentations. Then we get to temperature, which is another significant factor. We have a few more, and then we'll be finished up for today. So when we think about controlling our temperature, there's a couple different types of pathways and different ways we can control that. And thankfully, the monitoring of it is quite straightforward. It's a thermometer. But all microorganisms are going to have their own ideal temperature range. So this is why we would want to control or want to at least monitor the temperature. The ideal temperature for a typical fermentation is in that 20 to 24 degrees Celsius, which is quite warm in terms of maybe what people are seeing utilized. Anything below 20 degrees Celsius, our yeast and bacteria are going to have a relatively slow metabolism. Now, maybe we want that. Maybe we want that progression to be slower. That's just kind of the nature of those characteristics. If there's higher temperatures, we're going to have that kind of runaway fermentation that I mentioned, where it's like the roller coaster coming down a big hill. It's going to be difficult to stop at the place that we want it to stop at. And then also above 40 degrees Celsius, we start to worry about microorganisms dying. And so, again, that's probably not exactly the direction we're wanting to go, because then we're not going to have microorganisms present to be able to provide and develop those metabolites that we want. This note here about the longer the fermentation, the longer the time to produce metabolites and for those to enter the bean. Again, this can be a good or bad thing. I want to be cautious here. We're not necessarily saying that the best pathway forward is just to make all our fermentations 120-hour fermentations. That's not necessarily the best thing, the best direction to go, both practically, cost-wise, efficiency, etc. But the fact of the matter is, the longer those fermentations last, the more metabolites that are going to be produced. So understanding that some of these factors to control and extend fermentations we might want to encourage, but we have to do that within proper controls where we're not allowing negative characteristics to influence. Temperature can be very difficult to control in certain environments. So one of the things that we might look at is if we have, fermentation being this exothermic reaction, meaning that it's putting off heat. If we have a very cold environment, we might want to cover with a tarp or something to try to trap that heat to allow for that heat to warm up the environment slightly to facilitate more efficient coffee fermentation or fermentation reactions. If we're in a very hot environment, maybe we use water, or maybe we cover our tank so that we lower the temperature to reduce the impact of what that heat is going to cause. Even when you start your fermentations, if you're in a really hot environment, you probably don't want to start your fermentations at 10 a.m. in the morning, because you're going to go through some of the hottest times of the day, and it's going to really build up that progression of microorganisms. Maybe you would do it overnight, or maybe if you're a really cold area, maybe you want to do it during the hot part of the day to really leverage the heat that's being created in your environment to encourage microorganism development. There's not necessarily one way that's right or wrong. It's just understanding your context and then applying the principles to your context. This is an interesting study that looked at the growth of lactic acid bacteria at various temperatures. And you can see here 3 different lactic acid bacteria species. And you see that and I apologize if this is hard to see. I know somebody made a comment about that previously. I I've tried to enlarge the photos a little bit, but the here at the bottom you see this square which is 8 degrees Celsius. Here the bottom one is 4. In the middle, the bottom one is 4 degrees Celsius, and then on the right side, the bottom one is 4 degrees Celsius as well. So you can see that really as you start to get warmer from 8 to 12, 16, 20, 4, 8, 12, 16, 20, as you get warmer, you see that the growth of your microorganisms is much faster. So, I mean, we're talking here, this time is 100 hours, 200 hours, 300, 400 hours. So we're talking like a long time. But you can see how temperature is significant in impacting the growth progression of your microorganisms. Then we get to the factor of pH, and we're going to talk about pH and time, and then we're going to be done for today. But as I mentioned previously, we have different types of microorganisms that are going to either thrive or resist or die in certain levels of pH and certain levels of acidity or alkalinity. Traditionally, you find that your pH of a kind of traditional coffee fermentation starts around 5 to 6.5 and will decrease by around 2 to 2.5 points. So with inoculation, you may get something to go even below that to like 3.5, but that's typically what you find in your in kind of a traditional coffee fermentation. So bacteria will acidify the environment, but they will eventually begin to die off. Your lactic acid bacteria will be eventually refrained by their own production, meaning that lactic acid bacteria are going to produce lactic acid, which is going to drop the pH, and then eventually it creates an environment that they no longer can thrive in, and so therefore they begin to die off. And they also don't really handle large temperature increases. I mean, we're talking like upwards, very, very large. So the aspect, and we mentioned this when we talked about the mucilage breakdown, right? The acidification of that environment not only is going to contribute to the type of microorganisms that can thrive, but it's also going to impact the breakdown of your mucilage. So something that to keep in mind as well. If we have a very low pH, it's going to stimulate a very fast initial fermentation. We can utilize temperature. So if we raise the temperature, maybe we speed that up. Dry fermentation is going to also speed that up because it's going to, one, we don't have water to raise the pH, but we also are going to encourage that kind of heat absorption and kind of the speeding up of that fermentation rate. And we can also use an inoculant that would potentially speed up that fermentation rate to stimulate. If we want to increase the pH, we can add water. But again, remember, by doing that, you're going to dilute some of your sugars and even potentially some of the acidity because you're raising the pH in that system. So if that's why kind of really monitoring and looking at, and when we're talking about our fermentation rate, we're using pH to monitor that. And we're looking at how quickly that pH drops, and then does it level out, etc., as indication to us of what the microorganism activity is inside that system. This was an interesting study that looked at different types of inoculations. So here on the left-hand side, we have Intenso, SEMA, Oro, and then a control. On the right-hand side, we have Briosa, Oro, SEMA, Intensa, ADY1, and then a control. The names of these are just, they are commercial yeast products, particularly Lalamon's commercial yeast products. But the red in both of these pictures is the control. And so you can see, and this is tracking the pH progression or the pH drop. So you can see how with an inoculant, you have a much faster pH drop and then kind of you're leveling off. Likewise, you have the same with your kind of through a submerged. This one is a dry fermented on the left and then a washed or wet fermented submerged fermentation on the right. So you can see in both of those environments, inoculations are going to increase the speed of your decline in your pH, which makes sense because when you're introducing inoculants, you're introducing microorganisms that are going to produce those byproducts and lower your pH. So just an interesting example, an interesting study confirming that. Different ways that we're going to monitor your pH. Again, you can use pH meter, pH test paper. This was always an interesting one that I always thought it was a great idea. I've never utilized it. Taste is, if you want to begin to taste the pH change, I guess you can. I always get a little bit nervous about putting things in my mouth in a coffee fermenting environment. But we can always take the pH and those numbers, and we can then take that to the cupping table, and we can really begin to make some connections to resulting flavor quality and then kind of how that presents in the final cup. And then the last factor is time. And this is the last kind of factor we're looking at with controlling fermentations. The key aspect that we're looking at here is that if we want to encourage more development of metabolites and give those metabolites time to influence whether it is to actually penetrate the coffee seed, adhere to the surface, etc., we need to give those time. Here you have an example. This is a SEMA product, a commercial yeast product, and you can see over time the development of this 2-phenylethanol, which is a higher alcohol, typically attributed with having the aroma of rose. Your isoamyl acetate, which kind of has some fruity aromas, and then your butanol, which has kind of more green vegetal notes. But you can see the progression of time of these three different compounds up to 48 hours. And so you can see initially from 0 hours till about 24 hours, you see pretty rapid increase, huge jump at 28, and then a decline here at 32 hours. It's interesting that what I understand to be happening at this point is the secondary metabolism starting to happen or starting to take more of an influence. And so you have a decrease of some of these compounds because they're being utilized, maybe converted into other products, and then more is being produced. So I thought that was an interesting, I think you could rightly assume the increase in development of these compounds was this change here that was a little bit interesting to me to see, that they kind of dropped, especially your 2-phenylethanol dropped, but then continued to increase in production. So this is not saying, okay, everybody should ferment their coffee for 48 hours now. That's the way to do it. It's just showing you a result of how, in this example with this SEMA product, these were some of the compounds that were produced and at the levels that they were produced over time. Again, another example, this one talks about temperature. So this looks at SEMA at 15 degrees Celsius in a 15 degrees Celsius environment versus a 25 degrees Celsius environment. And you can see how a 25 degrees Celsius environment really begins to take off and expand in terms of the population expansion, kind of around this 23 hours or so, really begins to jump. And so the cooler the fermentation, the longer you can ferment. Okay, so the cooler the fermentation, the longer you can ferment, but your risk of stalling the fermentation. And Hutan, your question, I heard about chilled cherry process. They use nitrogen for controlling the tank temperature. Yeah, it's definitely possible. But the issue is that if you're going to ferment at a very low temperature like that, the risk that you run is stalling out the fermentation. And so it depends on, are you inoculating? Are you sure that the microorganisms that are present can thrive in a very low temperature environment? All of those factors are things that we need to consider as we're looking to control fermentation to achieve certain specific outcomes. So the point of kind of this lesson is identifying that there are certain key factors that we can utilize in controlling our fermentations. We can look at the initial population, we can look at food sources, oxygen levels, temperatures, pH, and time as all certain factors that go into producing certain results. So when we think about how do we track, how do we monitor, how do we keep data on our fermentations, each of these different aspects provides us different areas that we can utilize so that we can, take those data results and then we can correlate that to resulting flavors and resulting qualities. As fermentation occurs, will it generate its own heat, energy? Absolutely, 100%. And so that's something that we have to factor when we're thinking about temperature, thinking about time, and when we think about the various microorganisms that are in that system. If the temperature gets too hot, does that discourage certain microorganisms and then encourage other negative characteristics? Could potentially be. So how do we handle the heat that's being created? All right. Sorry for too much question. What's hot anaerobic? You're not. Please don't be sorry about asking too many questions. Hot anaerobic. I mean, again, it's so hard for me at times to say what a term is or isn't because I wasn't the one that said it, so I don't know what I was referring to. Or maybe if I did say hot anaerobic, I apologize, but I don't think I did. But I would assume what they mean is a low oxygen environment. So they've created a low oxygen environment through a tank, through something, and that they have added heat or that they've increased the heat. So they've increased the temperature to maybe speed up or to facilitate or encourage certain type of microorganisms in an anaerobic, in a low oxygen environment. So that's what I would assume that they mean. Is that positive? Is that negative? I could think of instances where that could be negative and produce some negative characteristics, or maybe other instances where it could be positive. Any other questions? We got about 15 minutes before I know we have people who need to jump off, and so wanted to give you an opportunity if you have any questions. The main things today are we're looking at how coffee fermentation has been used and defined in terms of its as a unit operation and kind of giving us a better context to understand that, and then looking at some of those different factors of how we control fermentation. Is there any research according using heat as byproduct of the fermentation? Yeah, so interestingly enough, so our Sukafina has some washing stations, several washing stations in Rwanda. And one of the things that they were looking at is the methane production from coffee from coffee fermenting. And even when you have your pulp, your skin sitting in the big, skin collection area, pulp collection area, the methane that's produced from that. So you could actually capture that methane and you can burn it as a fuel source. And actually by burning it, you convert it from methane to CO2, which reduces the greenhouse gas emissions significantly. Or you can actually convert that to a heat source that maybe it powers dryers, or maybe it also generates other types of potential for energy use. So yeah, it is something. Which hot would be considered above 24 Celsius up to 40 Celsius? Which hot could be considered above? Oh, which hot could be considered above? I would say hot, we kind of see a typical fermentation kind of in that 20 to 24 degrees Celsius. So as we get above 24, 25 degrees Celsius, we're starting to be concerned about things being a little too hot. But then once we get above 40 degrees Celsius, we really start being concerned about our microorganisms dying. No, it's okay, Tristan, no worries. I think I followed you. There's a question here. Can we say the longer the fermentation is, the higher alcohol it will be produced? And does it contribute to the more flavor we get? Okay, so your first question is the longer the fermentation is, the higher alcohol will be produced. If we have low oxygen and we have our yeast present, yes. And as long as we have sucrose or sugars, glucose available that they're consuming, that those yeasts are consuming, yes, we will continue to have alcohol fermentation, ethanol being produced. Does it contribute to the more flavor? That's the part that I can't really confirm. I can't say that just because we have more alcohol fermentation means that we have more flavor. It's not straightforward in that way, because not only is that ethanol, that alcohol is being produced, bringing about certain flavor characteristics, but it's also providing conversion of that ethanol. There's other microorganisms and other metabolic processes, sorry, that are creating esters and higher alcohols and ketones and other aromatic volatiles. So I can't really say that it does contribute. It definitely does contribute to flavor, but it's not necessarily okay. We just want to produce lots of alcohol so that we can have more flavor. It's not that straightforward. And where we're going to go tomorrow is linking some of the microbes that we have to actual flavors. So we'll walk through and we're gonna look at a couple different research studies that actually talk about specific microorganism species and their kind of what flavors and what characteristics they contribute. And so it'll be a little bit longer of a one lesson. We went through two different lessons today, but it'll be a little bit longer of one that will discuss that much more at length. All right. I am going to hang around for just a few more minutes if you have any other questions. But I will be sending you out another link for tomorrow afternoon. Yeah, thank you so much again for being flexible on time. And I look forward to seeing everybody tomorrow. Rest well and see you then.
See you. Thank you. Bye.
Enjoy your evening.